Okay, ladies and gentlemen, here we are for part 2 of cell parts, and let's take a look at ribosomes to start with. Okay, so ribosomes, these guys, their structure, 2 subunits made of protein and rRNA. There is no membrane here, so we see the small subunit and the large subunit. Their function is protein synthesis, so yes, they're small, but these guys are mighty. They are incredibly important. If we take a closer look here, you can see that ribosomes are found on the endoplasmic reticulum next to the nucleus there. They're also found what we call to be free floating within the cytoplasm or the cytosol. So we see here how they're attached to the ER right there, and then we see some free ones floating in the cytosol. Again, this is a TEM that's showing that picture. And if we want to look at a diagram, we're going to see the large portion and the small portion of the ribosome. So let's talk about those large and small portions. The large portion is composed of 45 proteins, 3 rRNA molecules. When we get to protein synthesis, you will understand rRNA and the significance of it, and especially in protein synthesis with that ribosome. The small ribosome is composed of only 23 proteins and only 1 rRNA molecule. Again, what you need to know, structure and function of that ribosome. Location of the ribosome. Again, free in the cytoplasm. They make proteins for use in the cytosol, and they are also membrane bound, which means that they are attached to the endoplasmic reticulum. Those that are membrane bound, attached to the ER, are going to make proteins that are going to be exported, usually from the cell. Okay, sometimes they still go within, but not usually. We're talking about going out of the cell. So 2 places, what you need to know, they are found in floating freely in the cytoplasm, and they are also found attached to the endoplasmic reticulum. Their job, make proteins. So let's take a look at the endomembrane system. Now the endomembrane system is characterized as a group of membranes that are related through direct physical continuity or by the transfer of membrane segments called vesicles. So this next part I want you to write down, but I wanted to show you the diagram of this. What is part of this endomembrane system? Well, check it out. I'm going to go way over here. Okay, here's what we have. Parts of the endomembrane system. Nuclear envelope. We have already talked about this. Smooth endoplasmic reticulum. The rough endoplasmic reticulum. Transport vesicles, which we will talk about. Golgi apparatus. Plasma membrane. Vacuoles and lysosomes. So we are going to focus on the endomembrane system today. So let's talk about the endoplasmic reticulum first. Oftentimes referred to as the ER. We will always write it as ER. You are more than welcome to write it as ER. You do not have to write it out every single time. The ER makes up more than half of the total membrane in cells, so it is a huge proportion of that endomembrane system. It is often continuous with the nuclear membrane. So when we build models of our cell, when we build that nucleus, we always put that endoplasmic reticulum, the ER, next to the nucleus, because it's continuous with that, and you'll see why when we talk about all the different functions and how everything is compartmentalized and works together. So the structure of the ER. It is composed of folded sheets or tubes of membranes called cisternae. So if you look at this diagram here, you see that the cisternae is pointing to the folded portion of the endoplasmic reticulum. Those red dots are ribosomes, and we'll talk about that a little bit more. When it says cisternal space, that's referring to the lumen. This is where some of the reactions are going to take place. But you need to know the cisternae. Again, structure of ER is very fluid, meaning that the membranes constantly change size and shape. It is not a fixed set organelle. This is a very fluid, it's going to change in how it appears. We'll talk about how and why. So types of the ER. Smooth ER does not have ribosomes. Its function, mainly used for lipid synthesis, carbohydrate storage, and detoxification of poisons. Rough ER has ribosomes, hence the term rough, because it has that studded appearance, so it's going to be caked with those ribosomes. Rough ER makes what we call secretory proteins, like for example insulin. These are wrapped in the membranes of vesicles, and they can be transported to other parts of the cell or out of the cell as well. The rough ER is also the membrane factory for the cell. What does that mean? It produces the membrane, that endomembrane system. It produces the membrane, and then that membrane becomes part of the endoplasmic reticulum. So it helps regenerate itself, so to speak. Now the Golgi apparatus is a second organelle that we're going to look at in the endomembrane system. Well, I say second, but hold on. The first one we covered was nuclear envelope. The second one we covered was endoplasmic reticulum, otherwise known as smooth and rough ER. And so now I guess we're on the third. I was thinking second just for today. So the Golgi apparatus. Let's talk about Golgi here. Golgi has a very distinct structure. It is a parallel array of flattened cisternae, and it looks like a stack of pita bread. There's about 3 to 20 Golgis per cell, and it is likely an outgrowth of the ER system. That is evolutionary. The idea is that the Golgi is an outgrowth. So let's look at its structure. Now this stack has distinct polarity, so we see a difference between the two sides, and so we identify the Golgi as having two poles. The cis face is what we refer to as a receiving side. This receives packages or vesicles from the ER. And the trans face is what we refer to as the shipping side of the Golgi, right here. And it forms vesicles and carries proteins to other sites. So I'm going to show you another big picture here. And here we go. So overall function of Golgis, processing. They modify the ER products. So the ER takes its products, packages it into a vesicle as part of that endomembrane system, packages it, and then sends it to the Golgi apparatus. The Golgi apparatus receives it on the cis face. Okay, so this is the part that it receives. Then it goes through, modifies that product from the ER, and then it distributes the ER product for transport, as you can see. So here's the trans face, new vesicle forming, boop, 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 shipping side, and it throws it out. And I don't know why, maybe it's the vesicle forming, but this always reminds me of old Star Wars, you know, where the spaceship comes in, where the spaceship comes in and lands, and this is the big space station, and then here's the spaceship, you know, leaving, being shipped out from the space station. That's how I always thought of it. Not saying that works for you, just what it looks like to me. Function. Now, what are those vesicles? They are small sacs of membranes that bed off the Golgi body, and they are the transportation vehicle for the modified ER products. Now, hold on, I don't want to get ahead of myself, so I'm trying to think of when I... Oh, this is extremely important. Okay. Those vesicles have ID tags. Think of it like on when you mail a letter, it's the zip code. It is what determines where that vesicle is going to get shipped. Now, what is an ID tag? It's not like it's your suitcase and you can wrap one of those little luggage tags around it and throw it out. We're talking about inside of a cell here. Remember those functional groups? We use functional groups like phosphates to tag them so that the cell knows where they're supposed to go. I just think that's amazing. Plus, the structure, the outside of that vesicle, might be structure specific to a place where it's supposed to go in the cell. So it recognizes its docking station and it knows that that's where it's supposed to be. So two different ways that vesicles can be targeted to go to the right spot. This is huge. It's not random. There is a system in place. Okay, so let's talk about lysosomes here. Lysosome is a membranous sac of hydrolytic enzymes. And these guys are made from the Golgi apparatus. So if we look in this, it's a single membrane here that surrounds that single wall membrane. And inside are a bunch of enzymes. Okay, so how do lysosomes come to be? Well, we started off with the protein synthesis in the rough ER. That got packaged, that vesicle went from the rough ER to the Golgi, and then the Golgi then modified it, transported it out from the trans side, and it's a lysosome. It's one of the things that a Golgi can make. So the function of a lysosome. Its function is to break down and degrade cellular materials. It contains enzymes for breakdowns of fats, proteins, polysaccharides, nucleic acids. It participates in two big things. These terms you gotta know. Phagocytosis engulfs food or invaders. For example, this is how amoebas eat. When the vacuole, which we'll talk about in a moment, takes in the food, then it bonds to the lysosome. The lysosome then digests the food, breaks down those monomers to be released for cell use. Or how white blood cells engulf bacteria. Okay, our white blood cells are like these big Pac-Men, right? Chomp, 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 chomp. White blood cells are really big lysosomes that go around and have those digestive enzymes. Chomp, chomp, chomp up the bad things. It's like Pac-Man eating up the ghosts. Autophagy is when a lysosome is used to recycle the cell's own organic material. So if we have an organelle that's broken down, it doesn't function anymore, that lysosome is going to ingest it and then break it down to recycle those monomers for cell use. So for example, here again we can see the rough ER forms a transport vesicle, goes to the cis face of the Golgi apparatus, modifies it, then the Golgi apparatus ships it out from the trans face here. Okay, and these are lysosomes in this scenario that it makes. Here is an example of autophagy where it's breaking, it looks like an old piece of mitochondria down. And this is one where the food or nutrients, and think those carbs from that donut I wish I would have ate, comes in, breaks off, and then the lysosome can digest the food in there. Again, another quick picture. This is a TEM of where we can see the lysosome in the nucleus. We can see here at high power the lysosome where it actually contains a peroxisome and a mitochondria, where the lysosome is going to attach and to start to break down those organelles to retrieve those monomers and put them back to be recycled for further cell use. And again, we can see digestion over here. So we got phagocytosis and autophagy. Huge, huge parts of lysosomes. Okay. Oh, wow. I'm running out of time. And this is it. Lysosomes are important in cell death. Missing enzymes may cause various genetic enzyme diseases referred to as lysosomal storage disease, and examples Tay-Sachs, Pompe's disease, and we're going to talk a lot about this in class and look at some examples.